So the modern system of classification involves naming with the help of numbering with the help of digits. So the Arabic numbers are used, and you can easily see examples I have mentioned here. For example, pepsin enzyme is classified as 3.4.23.1. That means its class will be third class, its subclass will be the fourth subclass, its subdivision is 23, and in that subdivision 23 the serial number first is occupied by. That means there are more enzymes apart from this in this subdivision. Same manner, trypsin is there. Trypsin is also in the same class, but its subdivision is also same, but its subclass is also same, but subdivision is 21, and then its serial number is 4. So every enzyme is nowadays in the modern system are given numerical digits for their identification. Now in your syllabus what you study is only the 6 classes, that's it. We don't study the subclass, subdivision and the serial numbers. So how many classes are there? And remember, you have to follow the same sequence which you are going to learn today. You cannot do it opposite, that any one class before and second class later. No, it has to be followed in this sequence because they are given numbers. This is the first class, second class, third class, fourth, fifth and sixth classes. So the first class of the enzymes is Oxido Reductase enzyme. That means it's a very easy one. Redox reaction you have learned in the 10th classes also. When oxygen is removed or there is addition of oxygen, or whether there is hydrogen getting removed or addition of the hydrogen, or the electron getting removed or the electron getting added. So these are redox reactions. You have learned these things even in the 10th classes also. When oxygen is added, it is called oxidation. When hydrogen and electrons are lost, this is also called oxidation. When oxygen is removed and H plus and hydrogens and electrons are added, this is called as reduction. You have done them in the 10th class. So accordingly the enzymes are called either as oxidase enzyme, reductase enzyme, catalase enzyme, peroxidase enzyme, dehydrogenase enzyme. So all these are various examples of enzymes which are in the category or in the first class, and the first class is oxido reductase enzyme. The second class is transferase enzyme. Transferase enzymes ability is but that if there are two substrates reacting and one of the substrate has a side group, this side group can easily be transferred from, so a bond is broken here and a bond is developed here without any change to these two molecules. This is called as transferase class of enzymes. And example, transmethylase, transaminase, transphosphorylase. So when methyl group is transferred, when amine group is transferred, when phosphate group is transferred. So accordingly it is called by various names. The third class of the enzymes is hydrolases. So remember, first is oxidoreductase, second is transferase, third is hydrolase enzyme. Now hydrolases are those which are involved in hydrolysis. I have already explained that to you. Hydrolysis, and the all digestive enzymes are hydrolysis enzymes, or you can say hydrolase enzyme. So whether you say nuclease enzyme, nucleotidase enzyme, nucleosidase enzyme, any one enzyme you take, lipase enzyme, pepsin, trypsin, chymotrypsin, all of them are hydrolases because they are involved in digestion, and digestion is a process which is also called as hydrolysis. Involvement of water is there. Now the fourth class of the enzymes is ligases. Ligases are those which also break down, so there will be a breakdown like the hydrolysis but without involvement of water. So there is no water involvement. For example, in your respiration chapter of the plant physiology, you will be learning about an aldolase enzyme. This enzyme breaks down 6-carbon sugar carrying 2-phosphate group, which is also called as fructose 1,6 biphosphate. So it's a fructose means 6-carbon sugar, biphosphate means 2 phosphates are there. It breaks into 2 compounds of 3 carbon and each carrying 1 phosphate. Those compound name are dihydroxy acetone phosphate and glyceraldehyde or phosphoglyceraldehyde. So one molecule is dihydroxy acetone phosphate and the second molecule is phosphoglyceraldehyde. So in this process there is no involvement of water, only breaking of a bigger molecule into smaller one. Example, aldolase enzyme. Next category is isomerase enzyme. So those enzymes which are involved in the process of isomerism or isomer formation are called as isomerase enzyme. Like for example, glucose 6-phosphate also carries 6 carbons and also carry 1 phosphate, can easily be isomerized into fructose 6-phosphate. So there is no change. The only thing is the arrangement of the molecules has changed, but everything else remains same. So it is called as isomerism. Example, isomerase enzyme or mutase enzyme, dismutase enzyme. In the same manner, the last class of the enzymes is ligases. Now, ligases are those which join smaller compounds to form bigger compound, also called as synthetase enzyme. So this is involved in the bond formation. Water may or may not come out in this process. Like for example, we have learned peptide bond formation, water comes out. But there are reactions in which water might not come out, and such enzymes are called ligase or synthetase. Example, synthase enzyme, like DNA synthase will join smaller pieces of DNA to form a longer piece of DNA. So these enzymes are named as synthase or synthetase. Example, ATP synthase enzyme, because ATP and phosphate will join to become ATP. So this is also joining of 2 molecules to form a bigger one. I hope this is clear.